This is Professor Doherty, and today I will be continuing on renal physiology. So this is going to be our second lecture on renal phys. And today what we're going to specifically focus on is water reabsorption. Water reabsorption is actually quite more complicated than what it sounds, at least in certain parts of the nephron. And then we'll take a look at electrolyte balance. That also can be quite complicated. And then we'll talk a little bit about renal acid-base balance. Now, all of these I want to emphasize are all connected to the blood, right? So when we reabsorb water, we're reabsorbing it from the filtrate back into the blood. When we talk about electrolyte balance, we're really talking about trying to maintain the balance in the blood, and the filtrate or urine is in fact kind of our dumping ground for extra electrolytes. And then same thing here for renal acid base. Basically our urine is going to be a dumping ground for free hydrogen in order to maintain our pH of our blood. So let's first talk about water reabsorption. So we already discussed in the last lecture that about 2/3 of our water was reabsorbed in the proximal convoluted tubule, if you recall that. So let me go over here to my whiteboard. So in the proximal convoluted tubule, we reabsorbed 2/3 of the water that came out in the filtrate. So if you remember, in the filtrate, we had about 180 liters that were leaving, right, or that were moved into the filtrate. So 180 liters of water per day, right, that is in the filtrate. We recovered in the proximal convoluted tubule. We recovered back into the blood, so I'm just using arrows to show that's going in the other direction, about 120 liters of water. And this was recovered through reabsorbance, right? So that means that we still have about another 60 liters that we have to deal with, that we're going to have to recover here. Okay, and so this is where we're going to talk about the recovery of at least some of that. So today, what I want to focus on right now for water recovery is what's called the countercurrent multiplier. The countercurrent multiplier occurs only in the loop of Henle. And it's a way of bringing water back into our blood. So our goal here is to try to make urine, or I should say try to make the filtrate hyperosmotic. So in other words, make the filtrate so it has a lot of solutes in it, or at least improve it to where it has more solutes in it. So let's go ahead and take a look at how this works. So I'm going to draw a nephron, because obviously this is taking place in the nephron. I'm going to draw a small glomerulus here, or I should say renal corpuscle, and a small distal convolute or proximal convolute tubule, and then a kind of wide loop of Henle, and then another small distal convolute tubule, and a collecting duct out over here. Okay, so here are all our pieces. I've just made the loop of Henle much larger here, and then I'm going to separate this off as well. I made the loop of Henle larger so that we could work with some numbers here around it. Okay, now if you recall, surrounding the loop of Henle there is the vasa recta, and that is our blood supply. I should draw this blood supply on top of the loop of Henle. So in other words, the vasa recta is going to be overlaying this. Okay. However, if I draw that, it's going to make our picture way too messy to understand. So instead what I'm going to do is I'm going to put a blood supply over here. This is how our blood flow is going to go. It really doesn't matter the direction. And essentially, this is a representation of the vasa recta. All right, so understand that this blood flow that I have over here to the side is really overlaying or next to the loop of Henle tubules. All right, so first of all, let's start off by talking about some numbers here. And what I want to first describe is the milliosmoles of solute that are in normal extracellular fluid. So in normal extracellular fluid, I'm going to write it over here so that it's not in the way. Extracellular fluid typically has a milliosmol content of about 300. So 300 milliosmoles would be the normal osmolarity for extracellular fluid. In other words, there's about 300 milliosmoles of stuff in that extracellular fluid water. That stuff includes things like sodium, potassium. So there's a number of things that actually make up this 300 milliosmoles. Sodium, potassium, chloride, glucose. There's a bunch of things. Normal, so that's normal extracellular fluid. Now in the filtrate, so I'm going to draw this in yellow here. So there's filtrate coming off of the glomerulus and off of the renal corpuscle. And as it moves through the proximal convoluted tubule, so here I'm using yellow arrows to depict that movement, it starts off at about a normal extracellular fluid concentration. So in other words, here the concentration of solutes in this fluid is about 300 milliosmoles. So in other words, the yellow arrows here representing filtrate that's inside of the tubules here has a concentration level of about 300 milliosmoles. Now, as this filtrate moves down, so I'm just going to draw these arrows here showing the movement. This is the movement of our filtrate. As it moves down the descending limb, so if you recall, this side is the descending limb on the loop of Henle, our concentration levels start to increase. And about here, it might be about 600 milliosmoles. So I'm just going to write 600, but you can write 600 milliosmoles. By the time we get to here, it might be about 1,000 milliosmoles. And by the time we get to the bottom down here, so again, I'm talking about the filtrate inside of the tubules here, it gets up to usually a maximum of about 1,200 to 1,400 milliosmoles by the time it's at the bottom of the loop of Henle. So in other words, it's becoming more and more concentrated as it moves down the descending limb of the loop of Henle. So remember, up here we were at 300, we're now at 1,400. So in other words, there's only two ways that this can happen, that we can have this rise in the amount of solutes in our solution. One way is to add more solutes to it, but that's not what's happening here. The other way to have this concentration increase is to remove the water and leave the solutes behind, and that's exactly what happens here. So what happens is on this side of the loop of Henle, so on the, let's see, I'll do this in black here, on the descending side, so on the descending side of the loop of Henle, this side, so this tubule here is permeable to water. Okay, so I'm going to highlight that here. So this whole side, this whole tube is permeable to water, meaning that it can allow water to flow out of it. And in fact, that's what happens. On this side of the loop of Henle, we're going to show this happening. So we have water exiting all the way down here. So water's coming out. So this is why our concentration is, or why our concentration of our filtrate is becoming more concentrated as we move down, is because water is being pulled out, but the solutes are being left behind. All right. Now, eventually this water is going to sweep over here into the blood, and so it's going to add to the blood plasma over here. And so it will get swept away. But now we need to describe why this water is willing to come out of this tubule. And the reason is because it's going to chase salt, so it's going to chase sodium. And that has to do with the ascending side of this limb. Okay, so let's right over here, the ascending part of the loop of Henle over here. This side of the loop of Henle has two things happening. First of all, it is actively transporting sodium, and chasing the sodium is going to be chloride. Let me remove some of this here. So sodium and chloride will chase it for free, but there is active transport of sodium. Just so you know, chloride is not being actively transported, but the sodium is, and the chloride will chase it. Also on the ascending side, there's something a little unique here, which is that this side is impermeable to water. So let me highlight that word, impermeable, meaning that water cannot get out of the filtrate on this side. And that is a very important point. So let me describe what happens here. As this filtrate, remember it's inside of the tubule, I'm just drawing the arrows on the outside to show what's happening here, or to show the flow. You can see here that as this pre-urine flows upwards here, what's actually happening here in the tubules, or in the cells of the tubules, I'm going to draw these very large, way oversized, is there are active sodium pumps in the cells in the tubules. Now this is a wildly inaccurate drawing, but I just want you to get an understanding of this. So these pumps are going to pump out sodium. So sodium gets pumped into the extracellular space. Now, for free, chloride also comes out of the filtrate. So the sodium is coming out of the filtrate and into the extracellular space here. Okay. For free, we also get chloride moving with it. So chloride is going to chase after all this sodium. I'm just going to put a couple of them here. All right. Now, if you recall, water likes to chase solutes. But here, remember that on this side, I'm going to put an X here. On this ascending limb of the loop of Henle, it is impermeable to water. So in other words, water cannot get out of the filtrate to chase after the sodium and the chloride. This causes this extracellular fluid to become very osmotic. So it's hyperosmotic out here. All right. The other thing that I want to emphasize is that, so even though it looks like in my drawing only this space is hyperosmotic, in fact the sodium and chloride is going to go everywhere. So it's going to make this hyperosmotic, it's going to make this hyperosmotic. All of this becomes hyperosmotic. All right, so the sodium chloride on the extracellular fluid is everywhere. It's not just in this center space. I want to emphasize that because what happens then is as this sodium and chloride basically kind of filters towards, so as it moves over towards the other limb over here, right, what it does is it draws out the water from the descending limb. And so that's what you're seeing over here. Okay, so this water from the descending limb is in fact chasing the solutes coming out of the ascending limb. And then together they all get swept away. So it's not just water that actually gets swept away in the vasa recta over here on the left. It's also the sodium and chloride as well. So if I drew them coming all the way across here, it would look very messy. So I'm just going to put sodium over here, also gets drawn in. The chloride from over there also gets drawn in. So all of this gets swept away in the blood. So we're basically reabsorbing all of this. We're reabsorbing the sodium, the chloride, and the water. But the big thing here is the reabsorption of the water, because that's the thing that's really moving in this loop of Henle. That's the thing that we're really drawing off here. Okay, Besides that, there is some contribution from urea as well. So you will see, let me pick a color here, urea will come out of the collecting duct over here, and it will also contribute to the osmolarity of the extracellular fluid to help draw off this water out of the descending limb. All right. The urea typically has a tendency to basically flow back into the loop of Henle after a while. So some of it will get taken up by the vasa recta, but it is contributing here, so I just want you to be aware of that. One other thing I would have you note too is the term countercurrent multiplier. Why is this called a countercurrent multiplier? Well, it's called countercurrent because the current of the filtrate flows against itself. So if you look on the descending side over here, see that this flow is going in this downward direction, and then if you look on the ascending side, the flow is going up in this direction. So these two flows are running countercurrent to each other. That's why this is called countercurrent. The multiplier is just used because you get this multiplier effect of sodium and chloride and urea all drawing water out of the descending limb. Now, one other thing I didn't draw on here are you get the same numbers on this ascending limb over here in terms of milliosmoles. So for instance, down here at the base, notice that our fluid, our filtrate, is at about 1400 milliosmoles, right? As it moves up, as the filtrate moves up through the ascending limb, it's going to become less osmotic. So we're going to have 1000 milliosmoles, and then it's going to go down to 600 milliosmoles. And then when it gets to the very top up here at the distal convoluted tubule, it will be somewhere between 100 and 300 milliosmoles. So in other words, it becomes less hyperosmotic. Okay, so it's almost basically at a normal extracellular fluid level when it gets to the other end here. Now you might ask yourself, well, what's the point of that? The point was to draw all this water off. So now we've drawn off significantly more water, and now we can continue to try to reconcentrate what's left of the filtrate. Now I like to use a analogy for myself to help myself understand the flow of the water here. So let me just give you this quick analogy that you can either use or not use. All right, so imagine that we have a location on the nephron here, and in that location we designate the amount of water in that area, okay, or that's flowing through those tubules. So my locations are going to flow like this. We're going to start at the proximal convoluted tubule, and then our water and our filtrate is going to flow to the loop of Henle. And then from the loop of Henle, that fluid is going to flow to the distal convoluted tubule and then eventually to the collecting duct. All right, so if we look at it like this, so imagine that the proximal convoluted tubule is a giant waterfall in terms of fluid that's flowing through it, okay? So proximal convoluted tubule is like the Niagara waterfall. It's huge. There's a ton of water there. And because that's what's coming off of the filtrate. But the proximal convoluted tubule gets rid of some of that water. It gets rid of about 2/3 of it, right? So our water flow then goes from being a giant waterfall down to a reasonable size river. So in the loop of Henle, we're now dealing with a river of water that's coming in. By the time the river gets to the end of the loop of Henle, we've drawn out enough water so it's now just down to a stream, maybe a good sized stream, and then the distal convoluted tubule is going to also get rid of some of that water, and eventually by the time we get to the end of the collecting duct, we're left with just a trickle of water left over. Okay, so you can see that we start with a ton of water in the system and we wind up with just a little bit. And two of the main places that we take care of that, the proximal convoluted tubule takes care of a huge amount of that. But then we have this complicated system in the loop of Henle that gets rid of quite a bit of it also, and then we get down to just a stream and a trickle at the end. Okay, so hopefully that helps you kind of understand how we're basically reducing and concentrating the water. So now I'd like to just show you a few slides that I have on this. And these just reinforce what I was just talking about. So we talked about the countercurrent multiplier. This slide here shows the vasa recta's role in the countercurrent multiplier. All right. I don't like overlaying my picture, so I did not draw all of this like the loop here. But you can see that even in the vasa recta, the blood of the vasa recta actually becomes more concentrated as well as it picks up that salt and urea. And then it becomes less concentrated as it begins to pick up some of the water that's flowing out of the filtrate. So there is this concentration gradient also in the vasa recta. I don't really require you to understand or know this as well as in the tubules. Now, this slide talks about urea. So I mentioned that urea is one of the solutes that water chases in order to come out of the loop of Henle and move into the vasa recta. This picture kind of shows this. So you should already have a good understanding that urea is our nitrogenous waste. It is in fact in our filtrate. Okay, it can get out of some of the tubules though, and the flow of urea is shown here in kind of this I don't know, beige color, I guess, or orange color. You can see here urea comes out of the collecting duct and moves into the extracellular fluid. This contributes to the hyperosmolarity of the extracellular fluid, allowing water to come out of the descending limb of the loop of Henle. Eventually this urea will be reabsorbed. You can see it being reabsorbed here. Or excuse me, I should say, well, now it's being secreted, but we're getting technical here. It basically, this urea will wind back up in the filtrate, most of it will, and then it will go out in the filtrate eventually. Now it shows here that this urea has to be excreted because it is toxic if we get too much of it. It is very permeable, meaning that it can move through these tubules, but like I say, it eventually will leave the body in urine. All right, this picture here, it's kind of a nice overlay. It shows the loop of Henle, so it basically shows a nephron here in yellow, and then the vasa recta laying next to it rather than on top of it. And you can see these numbers here represent the milliosmoles of osmolarity of either the filtrate that's inside of the nephron, or in the case of the vasa recta, it shows the numbers next to the vasa recta show the osmolarity of the blood. And you can see that these two basically kind of mirror each other. You don't have to really completely know these numbers on the vasa recta, but I do want you to know the numbers as they, at least the top and bottom numbers, as they move down and up the loop of Henle. That is kind of important. Now this picture also shows something that I didn't talk about before, or that I haven't gotten to yet, and that is if you look at the collecting duct is actually giving off water. So it's losing water as filtrate moves down the collecting duct, water is being shed from that filtrate and being picked up and back into the blood. Now if you recall over here on the loop of Henle, the descending side of the loop of Henle was permeable to water, the ascending side was impermeable to water, and now we have again a tubule that is permeable to water. However, this permeability is controlled, all right, and it is controlled by hormones. So this is a very important concept. Over here, everything in the loop of Henle, none of it was controlled by hormones. It just happens, okay? The filtration, or the pumping of the sodium and the leaving of the water, none of that is controlled in any significant way. Over here on the collecting duct, it is controlled, highly controlled by hormones. And here are the hormones. The first hormone is antidiuretic hormone. So ADH, you should be aware of, is an abbreviation for antidiuretic hormone. This hormone also has another name as well called vasopressin. So vasopressin and antidiuretic hormone are the same thing. This hormone is secreted by the pituitary gland, and it acts to increase water reabsorption. And where it acts is on the collecting tubules or collecting ducts of the kidney. So again, and here in this slide I have it saying the medulla collecting duct membrane is where this acts. So antidiuretic hormone acts on the medullary collecting duct. Normally, this collecting duct is impermeable to the high surrounding sodium chloride in the extracellular fluid. So in other words, under conditions where there's no antidiuretic hormone, then water is not going to move out of the filtrate in this region. Antidiuretic hormone makes the collecting duct cells permeable to water, and that's only in the presence of antidiuretic hormone. So if you think about it, what is a diuretic? A diuretic is something that makes you have to urinate. Antidiuretic hormone, this hormone, makes you pull water out of your pre-urine, making less urine, so it makes you have to pee less, right? Because it's pulling that water back into the body. How does antidiuretic hormone actually work down at the collecting duct? Here's what it does. Antidiuretic hormone binds to the collecting duct membrane receptor. So there are some receptors for antidiuretic hormone. In other words, these are cells that are lining the collecting duct, coming into contact with the filtrate. This activates the cAMP pathway. We've talked about cyclic AMP before. And this in turn winds up putting some proteins called aquaporins, and aquaporins are water channels. It puts them into what's called the apical membrane, so the top membrane of the cells of the collecting duct. Because of this, water then comes out of the filtrate by osmosis. So in other words, remember the surrounding extracellular tissue, it has a very high concentration of solutes. So water is going to chase solutes. And then that water eventually gets reabsorbed from the extracellular fluid into the blood plasma. And a secondary effect is that our filtrate becomes more concentrated. So let me see if I can just draw a quick picture of this for you. So imagine here, again, we have a nephron. And over here, I'm going to draw a collecting duct, but the collecting duct I'm going to draw very large. So this is our large collecting duct. And this collecting duct is the walls. Remember, if we really zoom in on it, the walls are lined with these cells. These are collecting duct membrane cells, essentially. So I'm drawing it super big, super blown up here, right? And remember that flowing in the lumen here is filtrate. So we have filtrate here flowing along, and it'll eventually become urine when it gets to the bottom of the collecting duct, but we're still not done with it. Now remember the extracellular fluid, remember there's extracellular fluid all around this collecting duct. And this extracellular fluid is hyperosmotic, meaning that there's a lot of stuff in it. So in other words, there's a lot of sodium. Remember, there's a bunch of sodium coming off over here. We have sodium being pumped out of over here. So we've got a bunch of sodium, and chloride is chasing that out. So we have a whole bunch of stuff, as well as there's some urea out here, and that's over here as well, right? So we've got all this stuff out here. So the water that's in the filtrate, so inside of here we have a whole bunch of water. And the water that's in the filtrate is hypoosmotic. So the filtrate is hypoosmotic. So in other words, there's a lot of water inside this tubule and not a whole lot outside. Normally these cells, so these are collecting duct cells, right? these cells normally won't allow water through. But when they receive a antidiuretic hormone, so let's say antidiuretic hormone comes down here from the pituitary gland, from the blood, this antidiuretic hormone will lock onto these cells, and it causes these cells to then place what are called aquaporins in their apical surface. So the apical surface here is facing the lumen. Remember that the lumen is the inside of the tube. So these green things, I'm going to draw a bunch of them here so I can label this. So these green things are aquaporins. And aquaporins are just channels that water can flow through. All right, they're just proteins. They make a tube. The tube is big enough for water to get through it, and that's about it. So when ADH locks onto the basilar side, the receptor side of these cells, it causes these aquaporins to what we call translocate, but basically move to the lumen surface over here, so the apical surface. When this happens, water from the filtrate will flow into the cells, and this is going to cause the cells to have too much water, and then that water will eventually flow into the extracellular fluid here. So all this water is just going to move through these cells over into the extracellular fluid. Now this extra water that's coming off will eventually go and be picked up by the blood supply, so it's going to wind back up as part of your blood plasma once it makes its way through the extracellular fluid. In that way, this filtrate is now becoming more concentrated, because now that the filtrate is losing all this water, it's eventually, its concentration levels are going to go back up, and eventually, if I remember correctly, it winds up at around, again, 1400 milliosmoles is the concentration usually of urine. So remember at the top up here, we were at about 300 milliosmoles, right? Whoops. 300 milliosmoles, and now we're at 1400 at the bottom of the collecting duct. So, you know, a decent amount of water can leave through the collecting duct. Now, antidiuretic hormone, just so you're aware, is always being produced by your body, and so you can slow it down, but you typically don't turn it off all the way. Some things that actually cause antidiuretic hormone to either not be produced or to slow down its production are anything that's a diuretic usually. So think about like coffee. Coffee is going to slow down the production of antidiuretic hormone, and what that means is that you're going to wind up with more fluid in your urine, which means more urine. You're going to have to urinate more. Now I'm going to tell you the irony of coffee in case anybody ever tells you coffee is bad for you because it's a diuretic. The fact is that when you drink coffee, you're drinking far more water in the coffee than what the caffeine causes to happen with antidiuretic hormone. So in other words, the amount of water that you pee out is not as much as you're drinking with coffee. So in fact, coffee does hydrate you. It just doesn't hydrate you as much as drinking something like water. So don't let somebody say that coffee dehydrates you, because technically it doesn't. Now the other thing that really turns off antidiuretic hormone and makes you have to pee as well is alcohol. So if you drink alcohol, if you've ever noticed, I'm sure most people have, if you drink alcohol, suddenly you have to pee quite a bit. A lot of people think that that's due to just the volume of fluid that you're drinking, and that is part of it, but a big part of it as well is that alcohol has a large effect on turning off antidiuretic hormone. So because of that, you're not absorbing in your kidneys as much water. So not only are you taking in fluid, but you're also then having less reabsorbed, and so therefore you have to pee more frequently. All right, this slide shows basically how water homeostasis works. I'll just step through a few things here to show you what they're talking about on this slide. This is from your book. Here we have someone who is dehydrated. They haven't been drinking enough water. What this leads to, and these are some concepts that you should understand, all right? So I want you to think about this. If you're dehydrated, this causes an increase in your blood plasma osmolality. So it causes an increase in your blood plasma osmolality. Why? So think about it. Your blood plasma has solutes in it. If you're not adding enough water to it, because you're always losing some water, right? If you're not adding water to it, it means that the osmolality or osmolarity, however you want to look at it, is going up because you have less water but the same amount of solutes. Okay. All right, so someone's dehydrated, blood plasma osmolality goes up. There are receptors in the hypothalamus that can read the dehydration levels of the blood. The hypothalamus then tells the pituitary gland, so we're going to be on this side of the line here, increase your production of antidiuretic hormone because we're low on blood, on water in our blood. Antidiuretic hormone production increases. This flows through the blood to the kidneys. The kidneys then increase their water reabsorption in the collecting duct, and this causes less water to be secreted in the urine or excreted in the urine. This is a negative feedback loop, right? I like how they colored this like a dark yellow to show you that your urine is getting darker. So that's when and that is actually why your urine is darker when you're dehydrated, is because you're reabsorbing more of the water, making your urine more concentrated. Now, if somebody overhydrates, so drinks too much water, now they're decreasing their plasma osmolality. That osmolality is read in the hypothalamus. The signal is transferred over to the pituitary. The hypothalamus tells the pituitary decrease the amount of antidiuretic hormone being produced. And it's actually the hypothalamus that's secreting this actually and releasing it through the pituitary. The decrease in antidiuretic hormone then goes to the kidneys, so it now has less antidiuretic hormone than what it normally would. This causes a decrease in water reabsorption, so you get more water coming out in your urine. And again, this is a negative feedback loop. So you're drinking too much water, it means more water comes out in the urine. We all understand this on an intuitive level because we've all done this before. You drink a lot of water, you have to pee a lot, is basically, but here you're actually seeing why. Now, here are some things that you should also understand here. Let's take a look at what happens in the blood, a stimulus from the blood, and the effect that it has on the secretion of antidiuretic hormone. If you have high osmolality of the blood, that's what this is talking about, which should indicate to you that someone is dehydrated. What is that going to do? You should understand, like for a test question, for instance, you should understand what that does to the secretion of ADH. In this case, because we have high blood osmolality or osmolarity, this is going to increase the secretion of ADH from the pituitary gland because, remember, this is going to cause more water to be kept at the kidneys. It's going to be kept in the blood, and that will offset the dehydration. The effect on the urine volume, though, is going to be to decrease it because we're not going to be peeing as much water out. And the effect over here, you can see increased water retention and decreased blood osmolality. If you have a decrease in osmolality, meaning you have too much water in the blood, you're going to decrease your antidiuretic hormone because you want to turn that off so more water leaves. This is going to cause an increase in your urine volume. Now, this can also be triggered by blood volume. So in other words, the release of antidiuretic hormone can also be triggered by blood volume, not just osmolality. If you have a large amount of blood volume, then what's going to happen is there are stretch receptors in your heart actually that will get stretched out. So imagine your blood volume is increased. This is then going to cause a decrease in the secretion of antidiuretic hormone, and that's going to increase your urine volume. And just the opposite is true as well. So if you have a decrease in blood volume, this is also going to affect the stretch receptors in the heart, and that's going to cause an increase in antidiuretic hormone production, and that's going to decrease urine volume. Should make sense, right? So if you have low blood volume, that means you're low on water in your blood. If you're low on water in your blood, then what do you think should happen with your urine? So you can basically just logically think these out, right? What should happen with your urine volume? Well, that should also go down because you don't want to be giving up a bunch of water while you need water in your blood. Okay, so that's a good way to think of these, is to just logically think them through. Next, I'd like to discuss electrolyte balance. So the kidneys play a critical role in electrolyte balance within the blood because of course they're tied to the blood very tightly. So let's talk about, first of all, what an electrolyte is, since some people aren't aware of this. So electrolytes are the following, and this is not an all-encompassing list. Electrolytes are pretty simple. They're just any ion that has a charge on it, essentially. So sodium, potassium are the two big ones, right? Hydrogen, chloride, bicarbonate, potassium, and calcium is another big one. There's some others as well, of course, like magnesium and stuff like that. But the two most important ones here for us are sodium and potassium. And of course, hydrogen is the big thing for pH. These are all important for the functioning of muscles, the functioning of nerves. We've discussed all of this before, as well as, in particular, sodium is really critical for blood pressure. It's a big regulator. So it regulates blood pressure and blood volume. And I don't want you to think that that's part of this here, so I'm going to erase that. There you go. All right, so all of these electrolytes are important to balance out, but the big ones are sodium and potassium, and maybe hydrogen as well for pH. So this kind of brings up something that if you've ever bought an electrolyte drink where they say, hey, buy this electrolyte drink, it will help to restore your electrolytes and do all this amazing stuff. Really, all that you're buying is sugar water, typically, and glucose can actually be one of those as well. But that's essentially what you're buying when you buy Gatorade, is expensive sugar water. All right, so the first thing I want to focus on then, since I'm talking about electrolyte balance, is sodium reabsorption. So in other words, that's sodium, right? Where does sodium reabsorption take place? I think in an earlier talk, I'll see if I can go back and correct it, I said that about 2/3 of it was reabsorbed in the proximal convoluted tubule. In fact, about 90% of it, so 90% of sodium is reabsorbed. So in other words, going from the filtrate into the blood, so it's reabsorbed in the proximal convoluted tubule. All right. Now, about 8% of it or so approximately 8%, whoops, so approximately 8% is reabsorbed in like the loop of Henle. Oops, keep screwing up my writing here. So in the loop of Henle, and again, this is reabsorption, right? And so this leaves us with about 2%, all right? The final 2% is going to be reabsorbed in the distal convoluted tubule. Now, I call it the distal convoluted tubule. The truth is that some of this reabsorption, the last 2%, does take place in the collecting duct, but I'm just going to pretend like it's all in the distal convoluted tubule to make it a little bit easier here. Now, here's the thing about this final 2%. This is very important here. The final 2% that's reabsorbed in the distal convoluted tubule. This is under hormonal control. And that hormone is aldosterone. All right. Now, aldosterone is produced in the adrenal cortex. And there are several methods by which aldosterone can be released. And I'll talk about at least one of those in just a moment. But aldosterone controls the last 2% of the reabsorption of sodium, like I mentioned. This final 2% comes out to be approximately 30 grams. So in other words, 30 grams of sodium final reabsorption is under the control of aldosterone. So it's about 30 grams per day. So that's a lot of sodium, actually. If you think about it, typical daily intake of sodium by the average person is about 3.5 grams per day. So, in other words, what I'm saying is that if you didn't have aldosterone to control that final 2%, you would lose 30 grams of sodium per day in your urine. Your intake is only 3.5 grams per day, so you would quickly perish due to the loss of sodium. So just to be clear, this aldosterone causes reabsorption of sodium. So in other words, it causes you to pull sodium, that last bit of sodium, those last 30 grams, from the filtrate back into your blood. All right. It will allow about the same amount that you eat out in your urine per day. So you will still lose sodium in your urine, and typically that's going to balance out the amount that you eat. So depending on how much sodium you eat, you're going to excrete the same amount or approximately the same amount. One other note I would make here, and I'm going to make it down here at the end, is that the sodium that's gained from the filtrate is exchanged for potassium or for hydrogen, for either one of those. But typically it's the potassium that it's exchanged for, so we'll start with that. In other words, potassium will move from the blood into the filtrate in exchange for getting sodium from the filtrate back into the blood. So there's basically an exchange. So this is an antiporter then, is how you would think of this, that we're getting an exchange of sodium for potassium through an antiporter pump where they're moving in opposite directions. Now, how do we get aldosterone to where we need it when we need it? Well, let's talk about aldosterone triggers here. So when I say aldosterone triggers, I'm talking about what causes aldosterone to be released. There are actually several things that cause aldosterone to be released, but one in particular that I want to talk about is the biggest one is a decrease in blood volume. If we have a decrease in blood volume, so let's say due to a lack of drinking water, for instance, what happens next? All right, so there's a decrease in blood volume. This decrease is detected in the kidneys at a place called the JG apparatus, and that is the juxtaglomerular apparatus, also known as the JG apparatus. This is a set of cells that sit between the afferent and efferent arterioles, and I'll show you a picture of this in a moment, or a drawing of this. These cells detect this drop in blood volume, all right, And when these cells detect this drop in blood volume, they then secrete renin. If you remember renin, renin is an enzyme. So renin is secreted. Renin is part of, remember the RAS system, I called it, right? So RAS, and that's what we're going to talk about here now. So this low blood volume triggers the RAS system. The first step in the RAS system is the secretion of renin. Renin, now remember, is an enzyme. This moves into the blood. So now we have renin in our blood. Also in our blood is a molecule called angiotensinogen. So angiotensinogen, oops. Angiotensinogen is also, so this is also in the blood, is an inactive enzyme. And the way that I know that is because at the end of the term it says ogen. Anytime you see ogen, that means that it's an inactive enzyme. Now what happens is renin is going to cleave angiotensinogen. And when it cleaves this, it cleaves it into angiotensin 1. Now, angiotensin 1 is an active enzyme itself, but it also gets cleaved, and it gets cleaved by an enzyme called ACE, which is angiotensin cleaving enzyme. So this ACE enzyme, I'm just going to leave it as ACE here, cleaves angiotensin 1 into angiotensin 2. Now, angiotensin 2 is also an active enzyme, and angiotensin 2 travels through the blood and it goes to the adrenal cortex, and it causes the release of aldosterone. Now, aldosterone is a hormone, and so because it's a hormone, it's also going to use the blood as a transport system. So all of these enzymes and hormones are in the blood. The only one that wasn't are aldosterone. Well, aldosterone and renin were not in there, but renin had to be secreted and aldosterone had to be secreted. Angiotensin was already in there, but it had to, in the blood, but it had to be cleaved. Now that we have angiotensin 2 causing the release of aldosterone into the blood, this aldosterone now goes back to the distal convoluted tubule. And let me get my drawing up here. And here, aldosterone triggers the cells of the distal convoluted tubule to reabsorb sodium in exchange for what we call the secretion of potassium, or under certain conditions it can instead use hydrogen. All right, so in other words, sodium is moving from the filtrate into the blood in exchange for potassium moving from the blood into the filtrate. All right, and that's basically the role of aldosterone and the RAS system. Understand, though, that the result of aldosterone bringing sodium back into the blood is that water is now going to follow the sodium. So water from the filtrate is now going to chase after that sodium, and this in turn is going to cause an increase in blood volume. So notice our starting and our end point here. So we have a start point of a decrease in blood volume, and our end point is a negative feedback loop of an increase in blood volume. So once again, aldosterone, when it causes sodium to move from the filtrate back into the blood, water from the filtrate chases that back into the blood and therefore increases the blood volume. All right, so let's take a look back on our slides here. So sodium reabsorption. This slide is basically just going over what I just talked about. So let's run through it real quick here. Imagine that you have in this case, a low intake of sodium. I used instead of low intake of water, but that's fine. So low intake of sodium. This winds up with a low amount or low plasma concentration of sodium. This triggers the hypothalamus to release or to decrease its release of antidiuretic hormone. The decrease in antidiuretic hormone causes a decrease in water reabsorption in the collecting ducts of the kidney. This increases urine volume, and this causes a decrease in blood volume. The decrease in blood volume is going to be detected by the JG apparatus. So this is basically where we were at. The JG apparatus begins to secrete renin. Renin goes into the blood. There's always angiotensinogen, I should say, in the blood. Through a series of cleaves, right, angiotensin 1 to angiotensin 2, we then wind up with angiotensin 2 in the blood. This goes to the adrenal cortex, causes the release of aldosterone. Aldosterone returns to the kidney through the blood and causes an increase in sodium reabsorption from the filtrate. This in turn causes an increase in sodium in the blood and therefore has a negative effect on this decrease in sodium to begin with. So negative feedback loop. Now here what they're not discussing is also the corollary return of water with the sodium, and that's going to change the blood volume. All right, so let's take a look at where renin is created. All right, so there are two types of cells here that I want you to be aware of that I'm going to talk about. So first of all, let's talk about where we're at in the body here. You can see here we have a very blown up view of the afferent arteriole leading into the glomerulus, and then here's the efferent arteriole on the other side leading out. There are distal convoluted tubule, I believe it is, or proximal convoluted tubule, one of the two, has their tubules that wrap up through this area next to the afferent and efferent arteriole. This area is called the JG apparatus. This JG apparatus is a set of cells that are between the afferent and efferent arteriole and smushed also between one of these tubules here. And you can see the cells here. There are two different types of cells that make up the JG apparatus. They're granular cells and macula densa cells. The granular cells produce renin. And so you should write that down. Granular cells of the JG apparatus produce renin. They do this based on baroreceptor feedback. So they're actually looking at blood pressure out here as well as blood volume in order to decide if they're going to produce renin. And of course, if we have low blood volume, low blood pressure, that is going to trigger these granular cells to produce renin, because that's going to then turn around and have the opposite effect to raise blood pressure and blood volume. The macula densa cells over here, they're actually looking at the filtrate that's traveling through the lumen of this convoluted tubule. What they do is they simply influence the granular cells, and basically depending on what's in the filtrate, they will influence the granular cells to possibly produce renin or possibly not. All right, so I just wanted you to be aware of where the JG apparatus is, and in particular the granular cells here. Next, let's take a look at some of the triggers for secreting aldosterone. All right, so I already talked about this one. This is the primary one. If we have a decrease in blood volume, which is typically due to a lack of water intake, think about what effect that's going to have downstream from there, okay, as far as the release of hormones and enzymes. If we have a decrease in blood volume, that's going to trigger those granular cells to increase their secretion of renin. That renin is then going to increase the amount of angiotensin 2 in the blood, and that in turn is going to increase the amount of aldosterone that's secreted. Next, what would happen if we had a high blood volume? Well, all three of these, as you would expect, become decreased. because here we have too much water in the blood essentially. And so therefore we want to get rid of some of that water. And we can get rid of some of that water simply by also getting rid of some of the extra sodium. Rather than bringing the sodium back in from the filtrate, we leave it in the filtrate, and that causes some of the water to then leave with it. Now, there are two other triggers here. One of them is important, the other one is not as important, but I do want you to know this trigger. So this is actually kind of interesting. So if you remember that when we brought sodium in from the filtrate, at the same time we secreted potassium. In other words, aldosterone also controls, or kind of has a secondary control on potassium. And so because of that, aldosterone can also be used to affect the potassium levels in the blood. This is not something that I always talk about, but I kind of have as a secondary thing here. Okay. So if you look here, if we have high blood potassium levels, that's what this is talking about. Remember, potassium does not trigger the granular cells, so it's not going to trigger the secretion of renin, so nothing happens here. Because we're not secreting more renin, we are now also not making more angiotensin, so that also does not change. But look over here, high levels of potassium will cause the secretion of aldosterone in higher levels in the adrenal cortex. So in other words, the adrenal cortex is sensitive to potassium levels, and if it sees that there are high potassium levels, it will cause the secretion of aldosterone, so that when aldosterone goes down to the kidneys, you're doing that sodium potassium exchange, and what that's actually doing now is pulling that extra potassium out of the blood. So then it becomes more about potassium. This is relatively rare, but it does happen sometimes. This condition, when this does happen when you have high blood potassium levels, is called hyperkalemia. So let me go ahead and write that over here. Let's go ahead and pull this down, clear this off. So hyperkalemia this means that you have high blood potassium levels. So too high. Now, a couple things about potassium here. So first, if you recall, 90% of our potassium was secreted, or excuse me, reabsorbed it is reabsorbed in the proximal convoluted tubule. So most of our potassium from the filtrate has already been reabsorbed. If you have no aldosterone, so if there's no aldosterone being secreted, this will lead to no potassium in the urine, because in the loop of Henle, so basically as you make your way through the loop of Henle and the last part of the loop of Henle in particular, you're actually reabsorbing the last 10% of potassium. So you'll have almost no potassium in your urine if you're not making aldosterone. It's actually a way that you can check for aldosterone production. And then the final thing is, remember that aldosterone can exchange for either potassium or hydrogen. So aldosterone can do either one of the following. It can either do this, so a sodium potassium exchange, or it can do this. It can do a sodium for hydrogen exchange. Now, if it does a sodium for hydrogen exchange, it typically will only do this when someone is in acidosis. So in other words, if you have very high levels of hydrogen in your blood due to metabolic acidosis or due to respiratory acidosis, then aldosterone will exchange, instead of potassium, it will exchange hydrogen instead, and it will dump hydrogen from the blood out into the urine in order to try to maintain the blood pH, because remember, blood pH is incredibly important, right? So yeah, this is a direct effector. It is pH, right? Now, this can actually lead to another problem, which is that if you have acidosis, and so aldosterone is taking this alternative pathway here, right, and doing this exchange rather than the exchange it's supposed to be doing, this can actually lead to a buildup of potassium in the blood because it's not being exchanged out like it's supposed to be being exchanged out into the urine. This buildup in potassium in the blood can lead to hyperkalemia. And in fact, this is something that you do have to watch for with people like diabetics. It's just another problem with diabetes. Remember that diabetics sometimes go into what's called metabolic acidosis because they're burning more fats than they should. This in turn can lead aldosterone to do a sodium hydrogen exchange instead of sodium potassium, and so in turn diabetics can wind up with hyperkalemia, so this high blood potassium level. Now, high blood potassium levels can lead to all sorts of misfunctioning in the muscles, in particular the cardiac cells, so you can wind up with all sorts of cardiac issues arising from this. All right, so coming back to our slide here, the fourth thing that can activate aldosterone is increased sympathetic nerve activity. This one I don't require you to know, so you don't need to know about this one. There is a hormone that has the opposite effect of aldosterone, and that is this hormone shown here, atrial natriuretic hormone, also known as atrial natriuretic peptide, or ANP. ANP actually comes from the cardiac muscle. It is released due to the stretch of the atria, and they will begin to release this hormone. Basically, the stretch, when the atria stretch out, it means that blood pressure is too high and we need some way to release that. And one way that we can relieve blood pressure is to reduce the blood volume, and that's what ANP is all about. So ANP opposes aldosterone, and so since aldosterone causes the reabsorption of sodium, ANP causes the release of sodium being kept in the urine essentially. And this, of course, sodium is going to draw water to it, so you're going to have more water also in the urine by keeping more sodium in the urine. So ANP is a hormone released by the heart. It is in direct opposition to aldosterone. Now, I just wanted to show this picture to show where potassium is reabsorbed and secreted. You can see here most of the potassium is reabsorbed in the proximal convoluted tubule. About 90% of the total is reabsorbed here. There's some reabsorption that takes place in the loop of Henle. You don't need to know about that. And then in the end, what happens though is you have secretion of potassium into the final product here, into the final filtrate, right before it goes into the collecting duct. And this is all due to hormonal control, and it's the hormonal control of aldosterone. Now, this picture is just a very simple picture showing the exchange of sodium for potassium, or potentially sodium for hydrogen, between the filtrate of the urine and the blood. So I'll just describe this real quickly. Basically, this yellow part down here, it represents the distal convoluted tubule, and possibly you can see here the part of the collecting duct and part of the loop of Henle that participates too, but I prefer just the distal convoluted tubule. You can see that within the distal convoluted tubule in the filtrate there's a bunch of sodium, and we can recapture this if aldosterone will come down to these cells and basically cause these cells along the edge of the tubule to pull sodium into the blood in exchange for potassium. They will sometimes exchange for hydrogen ions, but typically only if the blood is quite acidic. All right, and that brings us to the acid-base balance. And so remember, anytime we talk about acid-base balance, we're talking about the acid-base balance of the blood, unless we specifically say otherwise. All right, so in the kidneys, we actually excrete hydrogen. So in other words, your urine is going to be more acidic, typically under almost all conditions, um, than your blood. It just depends on what the acidity of the blood is. We also wind up reabsorbing most of the bicarbonate that comes out in the filtrate. So in other words, we're getting rid of hydrogen, we're trying to pull back in bicarbonate because bicarbonate acts as that nice buffer for us in the blood. Most of our hydrogen secretion, so remember this is moving hydrogen from the blood into the filtrate, occurs in the proximal convoluted tubule. And this is often in exchange for sodium. So remember we're trying to reabsorb sodium. So here we actually get a hydrogen sodium exchange, pulling sodium back into the blood. Remember we reabsorb about 90% of that sodium in the proximal convoluted tubule, and we'll move some hydrogen into the urine or into the filtrate at this point. Normal urine pH, as you can see here, is almost always slightly acidic, between 5 and 7. The goal is to maintain the blood pH at 7.4. In other words, your body really doesn't care what the pH of the urine is because we're going to get rid of it. What it cares about is the pH of the blood. So if the pH of the blood goes way off, then you're in deep trouble. But urine you can keep between this range, usually of 5 and 7. It's almost always acidic. Okay, so let's talk about how your body keeps bicarbonate. So here we're going to we're going to talk about bicarbonate reabsorption in the proximal convoluted tubule. So let's start off here with some cells of the proximal convoluted tubule. So here I'm going to draw some large cells of the proximal convoluted tubule. So these are the cells that are lining the tubule. Down here we're going to say that we have the lumen of the tubule is down here. So because of that we have filtrate flowing along this side. So this is our pre-urine. And over here we have a blood supply flowing. And of course I'm just eliminating all the extracellular fluid here, but that's fine. Okay, within the filtrate we have bicarbonate. So we're going to have HCO3 minus here, and we want to recover that into our blood because that's valuable. We also have in the filtrate a bunch of hydrogen as well. And this bicarbonate and this hydrogen will combine with each other in just a moment. One thing that I want to say though is that this bicarbonate cannot get through the walls of the proximal convoluted tubule. So if it tries to go through here, it won't be able to, and it'll get bounced back out. So the apical side of the proximal convoluted tubule cells is impermeable to bicarbonate. That's a problem for us. So what your body does is it puts what's called carbonic anhydrase. That's an enzyme that converts bicarbonate and hydrogen into, and it can go in either direction here, into carbonic acid. Now this carbonic acid is also going to convert into water and carbon dioxide. Now these two gases, or this gas and this fluid, can penetrate across the cell membrane. So these two are actually going to penetrate into the cell. And when they get into the cell, they are actually going to recombine. So it's kind of a tricky way to get bicarbonate into the cell is by having this carbonic anhydrase out here to convert it into oxygen and, or excuse me, water and carbon dioxide, and then those can move into the cell. Now, inside of the cell, there's also carbonic anhydrase, and that will reconvert these back into carbonic acid. Now, ignore the fact that I moved across the cell membrane. That means nothing here. All right, once this is converted back into carbonic acid, it can then split into bicarbonate and hydrogen ions. Now that the bicarbonate is on this side of the cell, or it's actually inside of the cell, as it begins to build up inside the cell, it is able to be transported out and into the blood, and that's what we wanted. So now we've recovered our bicarbonate and it can continue out with the blood. Now that leaves us with this hydrogen here inside of the cell, and we still, remember, your cells do not want this. We don't want this going back in the blood. And so you have some pumps. We'll put a pump over here. And remember that we have, do this one in red, we have sodium, extra sodium in the filtrate that we want to recover. And this pump here will pump the sodium into the cell, and at the same time it will move the hydrogen in exchange back out of the cell, so this hydrogen can now be swept away in our filtrate. It also keeps our urine a little more acidic. Now eventually the sodium is going to make its way up here into the blood, and so it'll be swept away as well as we recover it. So this is basically our recovery system for bicarbonate. Most of it takes place, if not all of it takes place, in the proximal convoluted tubule. So now let's go ahead and take a look at a few more facts about urine here. So let's talk a bit about urinary buffers. So the nephron cannot produce very acidic urine. So if it's below a pH of 4.5, it's typically just not going to happen. So very acidic urine it will damage the cells that are transporting it out, and so therefore we want to buffer that urine if we can. All right, and so that's what we're talking about here. It has to be bound to a cation, and there are really three buffers that are used. The first one we just talked about is bicarbonate, but the problem with bicarbonate is that we pull almost all of it out of the filtrate. Almost all of it winds up back in the blood. And so instead there are two other buffers. This is hydrogen phosphate, which can be used to buffer the hydrogen, or we can use ammonia to buffer the hydrogen, and then it just becomes ammonium, which would be NH4 plus. But this is also why your urine kind of smells, is because of this ammonia. But these are both buffers that are commonly used to keep your urine from becoming too acidic. Oops. So finally, let's go ahead and talk about diuretics. As you're probably aware from the name, diuretics increase the urine volume. All right, you can see that here. Now, diuretics can either be, something that you drink through just meals. For instance, like we talked about, coffee is a diuretic, alcohol is a diuretic. But these can also be drugs that are intentionally used as diuretics. So some of these drugs I want you to be familiar with here. So the first one, or the first set I should say, are loop diuretics. A couple of examples of these are Lasix and furosemide. Loop diuretics affect the loop of Henle, as you might imagine. And what they do is they inhibit sodium chloride transport out of the ascending limb of the loop of Henle. And here in particular, remember, what they're really inhibiting is the sodium transport, and then the chloride just follows that. So they block this transport. Thiazide diuretics, and here's an example. Thiazide diuretics inhibit sodium chloride reabsorption in the distal convoluted tubule. All right, so you can imagine that these are probably going to be offsetting or inhibiting the effects of aldosterone, right? Because really it's the sodium that we're concerned with here. The chloride will just follow the sodium. The next type of diuretic that we have are carbonic anhydrase inhibitors. Now, carbonic anhydrase inhibitors, so I just mentioned carbon anhydrase, which if you recall is used in the bicarbonate equation, right, to move it back and forth. Carbonic anhydrase inhibitors basically block carbonic anhydrase and therefore prevent water reabsorption when bicarbonate is reabsorbed in the proximal convoluted tubule. So If you think about it, so I didn't mention this when I mentioned that we're reabsorbing all the bicarbonate, but when we reabsorb bicarbonate from the filtrate, water is going to chase any solute, right? And so water will chase after that solute, so that prevents water reabsorption. And then the final one are osmotic diuretics. These include things like these types of sugars like mannitol that counteract glucose and diabetes mellitus. So for instance, somebody who is diabetic, part of the problem with being a diabetic is that you wind up with a bunch of glucose in your urine, and that glucose is then going to attract water to it. And so essentially you can use mannitol as a diuretic in order to offset this. If we look at where these diuretics actually affect, you can see here. So the furosemide diuretics are going to affect the sodium pumps on the ascending limb here. The thiazide diuretics are going to affect the sodium pumps on the distal convoluted tubule. And so they're basically going to offset aldosterone or possibly inhibit aldosterone. The carbonic anhydrase inhibitors are going to inhibit that process from happening here in the proximal convoluted tubule. All right, and with that, that is the end of this lecture. Thank you.